Hello. Due to the COVID-19 crisis, much was spoken about the PCR. The PCR is used like a diagnostic test to COVID-19. Due to this, I want to speak about this technique. PCR means polymerase chain reactions, and a term that can confuse is the protein C reactive. That is a protein used like an inflammation marker and is have no relation with the COVID-19. The PCR or polymerase chain reaction is a laboratory technique used to amplify DNA. Consists in three phases: the denaturation, the annealing, and the elongation, that are different by the temperature used and the process that occur in any one of these phases. We use this machine to make a PCR, the thermocycle, and we need these compounds. To make a PCR, we need the DNA to amplify, the primers that are short DNA fragments, DNA polymerase, DNTPs, and buffer with cofactors. The first phase is the denaturation. In that, we separate the DNA double strand with a high temperature, like 95 degrees Celsius. The next phase is the annealing. In that, with a minor temperature, like 50 degrees Celsius, we permit that the primers joins to the DNA strands. The other phase is the elongation. In that, with a temperature close to 60 degrees Celsius, in this phase, the polymerase joins DNTPase to the DNA strands, making a copy of the DNA. These three phases are repeated in various cycles, amplifying the DNA. But how we can visualize this DNA? We can visualize the DNA making an electrophoresis. In the electrophoresis, we put the amplifier DNA in an agar gel and connect to voltage. The DNA migrates along the agar gel due to his weight, and we can identify our DNA for his size. Other kind of PCR usually used are the RT-PCR or reverse transcriptase PCR. In that, we use an enzyme called reverse transcriptase to convert RNA in DNA and later amplify with PCR. The other PCR usually used is the qPCR or quantitative PCR. In that, we use a fluorescent marker to measure the DNA quantity in a sample. We obtain this kind of graph in this kind of PCR. In the diagnostic of COVID-19, is used a combination of this PCR. The RT-PCR is used to convert the viral RNA in DNA, and the qPCR is used to amplify and measure this DNA.